So yes, thank you. Thank you for the opportunity to talk here, and I'm actually very happy to be following Hongyang's very excellent talk. I think it builds just a perfect bridge from the beautiful talks about metabolism and metabolic pathways this morning towards the ways in which I would like to think a little bit about metabolism. And I should mention that I'm going to be talking about a story, an evolutionary story that plays out much more recent than origins, so several billion years later. But I think it's very relevant because, of course, if there are physical laws, physical principles that are governing the emergence of life, they don't go away once life is there, right? And so studying evolution in intractable systems that are playing out more recently can actually help us think about what those principles are. But in doing that, I think it's important for us to think about finding the correct sort of reference frames to be thinking about evolution that are compatible to asking how we ask questions about emergence. And so, in Hongyang's talk focused on ideas of information flow and how that relates to population dynamics. And I want to sort of talk a little bit more about mass and energy flows and sort of at that level. So, in my work in general, I'm interested in metabolic evolution. I study metabolism in general. And one of the most interesting things actually about metabolism to me is that it's not just a system, but it's actually also a lens through which we can study organisms. And it's because metabolism expresses itself at many different scales within living systems. So at the lowest level, metabolism is basically chemical reactions like organic chemistry, catalyzed by small metals and enzymes and cofactors. If we zoom out to the cellular level, metabolism is now networks of that kind of chemistry that cells use to take in their environmental inputs and recreate themselves. If you keep zooming out to the level of ecosystem, metabolism now has networks of that kind of networks, where it mediates not just the growth of cells, but also the interactions, the social interactions, and the trophic exchanges between cells. And if you keep zooming out, at the global scale, the metabolic activity of all cells on Earth together is what's mediating the global elemental cycles. But the key point is that all of these scales is the same chemistry, right? And so metabolism provides kind of a lens to be thinking about how we link up things like pathway innovations, to adaptive radiations, to ecosystem restructuring, and to global biogeochemical perturbations. And the other thing in there is that because we're thinking at the level of chemistry, again, and this comes back to sort of some of the other themes we've been talking about before, it allows us to think about metabolic or evolutionary change in terms of shifts between regimes of chemical dynamics, and sort of in sort of a physical way of thinking about evolution in that sense. The system I'll be talking about in trying to build up some of these ideas, was introduced by Hongyang. It's this cyanobacteria that lives in the surface oceans called Prochlorococcus. It's one of the most abundant cell living organisms on Earth. It lives between sort of these bands here, everywhere out in the open ocean, and forms sort of a planet-wrapping population of cells. There's several times 10 to the 27 of these cells on Earth, and they form a sizable fraction of primary productivity and photosynthesis on Earth. They're part of what's known as the marine picocyanobacteria, where together with their sister lineage, marine Synechococcus, they fix something like a quarter of all CO2 in the oceans. So really it's a cellular system, but it's also a planetary system, right? And so when we think about the evolution of this system, it's also interesting to sort of think about and keep in mind what happens to the planet when the system switches on and comes online in Earth history. So there's several things about the system that make it very appealing for studying evolution and for studying metabolism. One of the things is that, as I mentioned, it has very ways in which its ecology and the relationship between ecology and phylogeny are very well understood. So if you go out into the open ocean and you look by depth, what you'll see is that the population separates into sort of layered population structure, where the sort of deepest branching populations live at the bottom of the euphotic zone, where light energy is low and nutrient concentrations are elevated, whereas the most recent branching organisms, lineages, live at the top, where light energy is abundant, nutrient concentrations are low. Right? So you have this sort of central light-nutrient gradient that organizes the system. And so what I set out to do is to see how does the metabolism of this system change along that gradient, and what does that tell us about the evolution of the system as a whole. And a lot of what I'm talking about, is in, if you want to read about it, is in this paper that came out earlier this past year. Right, and so to look at this question, I'm thankful to Donato for already introducing this idea earlier. So to do that, I've used this system which I've been trying to push now for the last several years, where basically you integrate metabolic network reconstructions with phylogenetic reconstructions to build trees of functional metabolic networks. So the way this works is, let's say you have some network that you're interested in, where these nodes are molecules and the links are reactions, and in black are all the parts that are universal, so everyone has them. And let's say that you know that within that clade or in that group you're looking at, there's 3 known alternatives, a pink pathway, a yellow pathway, and a blue pathway. What you can then do is take all the genomes in that clade or in that phylum and look at the distribution of the genes encoding those pathways. And so let's say that in this case, you see that the genes for the yellow pathway are universally distributed across your tree and across the roots, and that implies that the yellow pathway is the ancestral pathway, and that pink and blue are derived from it. There's one additional key constraint, which is that in going from yellow to, say, blue, you have to first gain the blue pathway before you can lose the yellow pathway, because if you were to first lose the yellow pathway, flow through the network is cut and the lineage dies off, right? And so what this then means is that you have now a tree of phenotypes. So these are all functional metabolic networks. What you can then do is you can start looking at the biochemical properties of the individual pathways to try to understand what drove the system from one state to another one. So for example, looking at differences in trace metal usages of the enzymes, maybe there's oxygen sensitivities in the different enzymes, maybe there's differences in ATP usages between different pathways. And by making these kind of comparisons, you can start to infer what was the evolutionary driving mechanism. So taking this kind of approach and looking at Prochlorococcus, you see something very dramatic actually happens. So what I've shown here is the metabolic core. So this is the photosynthetic electron transport chain and this core carbohydrate metabolism. This is where electrons and CO2 come together to initiate all of biosynthesis. And what happens actually in this part of the network is that you see that Prochlorococcus forms a very unique lineages within the rest of cyanobacteria. The whole metabolic core undergoes remodeling in a way that is uncommon in the rest of cyanobacteria. So almost everything undergoes change. You see genes are being lost, genes are being gained, the stoichiometries and the pigments of the photosystems are undergoing change. So a lot is happening, which already is telling you a lot, right? Because this is where everything starts, so change is not going to be easy. So if you see change, it's suggesting of a strong driving force. And the other thing that we see as part of this change, sort of surprisingly initially, is that it seems to be adding what look like electron drains in the network, right? So as the system is evolving, networks are being interrupted, and it looks like pathways are being added for excreting small organic carbon molecules out of the cell. Now that seems kind of like a surprising thing. Why would a system be evolving to burn energy out into the environment in that sense? And so to try to make sense of this, I took the information that we know from metabolism and tried to integrate it with everything else we know about the system. So looking at the population structure, genome evolution, proteome evolution, growth rate, light physiology, everything else that we can find. And when you do that, when you integrate all that information together, it turns out that you can actually align all of the macro scale diversity within this system along a single physiological axis, which is suggesting that over geologic time, the system evolution is sort of steadily maximizing this one single physiological parameter, which is the ratio of electron flux to the nutrient flux in the cell. So let me unpack that a little bit. Electron flux is just light intensity times absorption cross-section times quantum efficiency, and nutrient flux is the growth rate times the nutrient quota, the number of atoms in a cell. And so, I already mentioned that you see this light level differentiation here. So these see more light than these. So that's, driving an increase in electron flux. But at the same time, the photosynthetic machinery is also undergoing change in a way that it increases the absorption cross-section of the cells. So the stoichiometry and the pigments of the photosystems are being modified in such a way that they're also absorbing more of the light that they see, of the blue wavelength light. And at the same time, we see that the nutrient flux is steadily going down in this lineage. So you see that the cells are getting smaller, the membranes are going from phospholipid to sulfolipids, the genome and the proteome is streamlined in ways that save nitrogen and phosphorus as well. And the changes to the photostatic machinery and the metabolic network end up saving iron as well. So what in fact we were seeing in the addition of these carbon excretion pathways is reflective of a more general process, which is increasing the through flow of electrons through the system, and sort of as a byproduct of that, then ends up excreting some of these electrons into the environment. Now try to ask what that means. right? So what electrons do is they transfer energy from the environment into the cell. So in fact, what this is suggesting is that what's going on here is that evolution is steadily increasing metabolic rate of the cell. And so the question then becomes, why should evolution steadily increase metabolic rate and excretion of organic carbon? And so there's a couple of additional clues about the system that will help us understand why this might be. And the main one here is that this highest metabolic rate phenotype actually lives in the highest surface layers environment where the light energy is most abundant and the nutrient concentrations are the lowest, right? So it suggests that there's a link between metabolic rate and nutrient acquisition. And that's basically what we ended up arguing in the paper, which is that what's going on here is that evolution is producing an ever more powerful vacuum cleaner, if you will, for sucking up the recalcitrant nutrients from the environment, and as a sort of byproduct of that, a sort of exhaust of this more powerful engine excreting small organic molecules into the environment. We formalized that a little bit more by taking reversible Michaelis-Menten enzyme kinetics and rewriting it in terms of nutrient concentrations. And for those of you familiar with resource ratio theory from ecology, it's sort of an extension of that to geologic time. And when you do that then, so now you see some of the terms we saw before. The nutrient flux is the electron flux. This is the efficiency of carbon fixation, which then gives you the carbon flux, the fraction of excreted carbon here in beta, and the delta G here is the free energy of the nutrient uptake reaction. And what you can then do is ask if selection is acting now on the nutrient concentration of the population at which they live, favoring populations that can grow at lower nutrient concentrations, what sort of cellular features will start to emerge? And so you can see very quickly that what you would want to do is you would lower your nutrient flux, which is what we see happening in the system. But a key point here is that as the nutrient concentrations drop and cells figure out a way to grow at lower nutrient concentrations, the free energy cost of the nutrient uptake reaction actually goes up. So it becomes more thermodynamically uphill. And so basically what I think is a good way of thinking about what the system is then telling us is that the way it sort of solves that problem is by using these excess electrons to drive the system into saturation and coupling that saturated state metabolism to that uphill nutrient uptake reaction. But that brings along with it a lot of extra carbon that the cell doesn't need, and in fact it wants less of it. And so the excreted carbon allows it to keep increasing this term up here without increasing the overall parameter, because it's actually counterproductive to increase this term. So if you increase both the electron flux and the excreted carbon fraction, you can actually solve the whole problem then. So what are the implications of this framework? Well, it has a few things, and the first thing that it says is that if you look at this population structure that we see in the extant oceans, the implication is that the way that came about is that you had a sequence of innovations near the surface where an ancestral ecotype was living in steady state equilibrium with its environment. An innovation comes in that enhances the energy flow through the system. That begins to draw down the nutrient concentrations in the surface, and then allowing a new type to start expanding in. And as the nutrient concentrations drop, this ancestral ecotype, which was adapted to higher nutrient concentrations, becomes restricted further down the water column. So basically evolution is sort of steadily bending this nutrient curve further and further down and compressing this population structure further and further down the water column. What this also means then is that the implication of this is that Prochlorococcus and other phytoplankton as well have an important role in shaping the chemistry in the extant oceans. And indeed, if you look at Prochlorococcus distributions, what you will see, and this is from a transect here of a ship going across the whole Atlantic, is that, and this is depth on this axis here, what you'll see is that the Prochlorococcus populations sort of retrace the nutrient minima, right? So everywhere Prochlorococcus is, the nutrient concentrations are lowest. But it turns out that that's also the environment where the dissolved organic carbon levels are at their highest. So dissolved organic carbon in the global ocean is everywhere sort of at around 40 micromolar, except exactly in these subtropical surface oceans where the concentration spikes up to 80 or 100 micromolar. Now, this, of course, has all kinds of implications for evolution at the ecosystem level, right? Both the drawdown of the nutrients drives evolution in others, but also the excretion of organic carbon. And to look at the impacts of that secondary step, I looked at what happens in the metabolism of SAR11, which is the most abundant heterotroph on the planet, co-occurs with Prochlorococcus in these environments, and it's in fact almost a little bit more abundant Prochlorococcus globally, but they co-occur always in the same environment. And if you do this kind of same sort of analysis that I did for Prochlorococcus for SAR11, what you'll see that again, its metabolic core undergoes a global reorganization, pathways being gained and lost. And if you map that diversity onto its phylogeny, what you'll see is that the metabolic core changes in a way that's complementary to that of Prochlorococcus. So all the pathways that emerge as excretion pathways in Prochlorococcus emerge as uptake pathways in SAR11. And there's also an excretion pathway that emerges in SAR11 matching an uptake pathway in Prochlorococcus. So you have kind of an emergent sort of free-living mutualism between the two that you can start to see. But it even gets a little bit more interesting than that, because if you look at the metabolic organization in this system and how Prochlorococcus and SAR11 interact, and you compare that with how metabolism is distributed across the organelles of plant cells, what you'll see is that they look like almost exact analogs of one another. So up here is the ocean microbe community, and down here are plant cells. And what you'll see is that basically the same pathways that are being used for electron exchange between Prochlorococcus and SAR11 look very similar to the pathways that chloroplasts and mitochondria use to exchange electrons. So in both cases, we see intermediates of lower glycolysis and photorespiration sending electrons from the autotrophic to the heterotrophic side. And in both cases, intermediates from the citric acid cycle allow electron flow in the opposite direction. But there are a lot more other similarities beyond that. So for example, it's known in ocean communities that Prochlorococcus and some SAR11 have given up the ability to detoxify peroxide, which is a byproduct of photosynthesis, relying on co-occurring heterotrophs that do that function for them. Same thing in plant cells. The chloroplasts and the mitochondria don't do the peroxidase reaction, the peroxide detoxification reaction itself. That's done by the peroxisome. If you look at other enzymes or other genes within the photostatic electron transport chains, of actually the electron transport chains on both sides of the equation, you see that the same genes are being added to the electron transports in both systems that are draining electrons from the electron transport chain. In both cases, the same genes are being added. If you look at the photosystems themselves and the photostatic electron transport chains themselves, you see Prochlorococcus is somewhat unique among cyanobacteria for having chlorophyll A and B. It has a tweaked stoichiometry of its photosystem 2 to 1. It uses different pigments, in chlorophyll A and B, as I mentioned. And all those features are similar to what plant chloroplasts do as well. So if you zoom out then to this kind of eagle-eye view of this and focusing on the metabolism, it really kind of starts to look as if the global ocean in the tropical and subtropical surface oceans, these communities are essentially free-floating chloroplasts and mitochondria sort of interacting with each other just like organelles of plant cells do. Or alternatively, that a plant cell is like a microscopic microbial ocean recapitulated in each cell. But I think it also allows us to think about mutualism in a slightly different way, because what these electron exchange pathways are doing is that what was initially otherwise going to be metabolic waste, energetic waste, now these exchange pathways are essentially recycling those electrons, allowing the maximization of electron flux to proceed now at the community level, whereas the loss of these peroxide detoxification genes and the abundant components of the system and restricting them to the less abundant ones, and the key point here is that these genes involve iron in their active site, lowers the average iron flux of the components. And so what I think this is saying is that in many ways mutualisms are sort of like a self-amplifying feedback loop that maximize the electron to nutrient flux at the ecosystem level, sort of emerging from that same drive operating at the lower level of organization. So kind of coming back then sort of and zooming out to a sort of more general level, what I think this is telling us then is that at the ecosystem level, the way evolution operates is that ecosystems as a whole follow these sort of self-amplifying and self-organizing evolutionary trajectories, where innovations that increase the energy input into the system cause a drawdown of nutrients in the environment and an increase in the biomass standing stock of the ecosystem. But this is done by increasing the electron flux faster than the nutrient flux, and so you have an increased amount of excess electrons sort of circulating through the system, and those excess electrons then drive the organization and the emergence of higher levels of organization that continue that same sort of process. And so the key point then is that if you look, I think what this is saying is that if you look at evolution on geologic timescales, it's not actually a zero-sum game. Instead, what it's doing is it's promoting a more energetic, a more massive, and a more complex biosphere. Now, this has then implications for thinking about Earth history as a whole. So if you think about where oxygen in our atmosphere comes from, This is sort of a simplified view of the carbon cycle, photosynthesis balanced against respiration. Now, of course, this is ultimately the source of the oxygen, but if these two processes are perfectly balanced, you won't actually have free oxygen. So, over long time scales of history, it's thought that the way we accumulate atmosphere in the oxygen is that a small fraction of that carbon ends up being buried inside of the Earth's crust. So if you now think about this, on top of this kind of evolutionary dynamic where you have these innovations that increase the global standing stock of organic carbon, and you have sort of always a steady background burial rate of 0.1% or so, if you suddenly increase the total standing stock for organic carbon, then this relative flux increases the total absolute flux of organic carbon burial. Now, so it turns out that if you look at molecular clock analyses, that Prochlorococcus and the marine picocyanobacteria, and in fact all planktonic cyanobacteria, emerge sort of right around this time in Earth history here, between 500 million and about 1 billion years ago, when oxygen starts to rise in the atmosphere. So down here, just a little bit more general point, this is sort of like a general view of the history of Earth oxygenation, and a lot of people often focus on the Great Oxidation Event here, which is when cyanobacteria are thought to have driven or been part of the driving mechanism of oxygenating the Earth initially. But at least as interesting to me is the secondary rise of oxygen here at this period, where oxygen goes from maybe about 1% up to, starts to rise up towards modern levels. And that's exactly around the time that these marine lineages start to emerge. So the question then is, what can these lineages teach us about the oxygenation of Earth beyond just increasing standing stock of organic carbon? And to think about this, it's useful to kind of briefly review what we know about ocean chemistry at that time. I should say this is very hard to study, and this is very hotly debated still, but there appears to be at least some consensus that in the deep open oceans, at least the deeper part of the water column remained anoxic well after the GOE, dominated by iron 2+, and maybe sort of at these continental margins where there was higher productivity. Maybe there was what's known as eosinia, driving abundant sulfide ions. And whereas in the modern oceans, they're largely oxygenated. And so the question is, what controls this transition? People have suggested several different negative feedbacks. One is that the sulfide ions can react with molybdenum, and sort of like, because molybdenum has a central role in much of the nitrogen cycle, can potentially impose some kind of nitrogen limitation on ocean primary productivity. Other people have talked about how iron 2+ can react with phosphate as a sort of a grip on the phosphate, imposing phosphorus limitation on the ocean biosphere. But the point is, to get over both of those negative feedbacks, you need to oxygenate the system, right? So to oxygenate, you have to oxygenate, which is kind of a hard thing to think about. And so people have often argued or thought about what kind of perturbations might allow you to get past that system. But what I like to think about is whether there are other feedback loops that may allow us to think about this in a more deterministic way. And what I would like to pose is that iron actually provides such a mechanism. And the way to think about this is that after this period in Earth history, you start to see that indeed molybdenum and phosphorus start to rise in the ocean. The concentrations start to rise, which is good for primary productivity. But iron actually drops by a few orders of magnitude. And that's actually a problem because iron is central to photosynthesis. Right? So if you're a photosynthetic cell, you have to figure out a way first to deal with a drop in iron before you can do photosynthesis in that dropped iron environment. So to say that another way, I think that ancestral oxygenic photosynthesis inherently has within it a self-negative feedback loop, where this photosynthetic process here depends on iron, and therefore depends on a flux of iron into the system, but it produces a flux of oxygen which sort of ends up extinguishing this flux of iron here. So you have to figure out a way to deal with this negative feedback loop. And if you look at what extant phytoplankton do and Prochlorococcus do, they do several things. One is that they modify the stoichiometry of the system to minimize the usage of iron. And what they do is also they enhance the absorption cross-section of these systems to make it more efficient, which allows more oxygen production with less iron. But the other thing that it does is that in the ocean today, iron is actually not free. It's bound to dissolved organic carbon, which is exactly this, and in fact the sorts of molecules that are being produced by Prochlorococcus, these carboxylic acids. So what you then have is, within the metabolic evolution that we're sort of seeing in Prochlorococcus, is built in now a positive feedback loop, where the increase of energy flow through the system is producing as a byproduct these ligands which can bind iron and enhance their solubility and accessibility within this oxygenated environment. And moreover, this positive feedback loop is related to metabolic rate. So as the energy flow, as the metabolic rate increases, this positive feedback loop gets strengthened, and you can increasingly start to overcome this negative feedback loop. So to summarize then, you have these phytoplankton here which produce VOC, which bind the iron to stabilize its abundance. But it's actually a secondary thing, which is that those same iron ligands are known to be able to, in other systems, be able to dissolve rock minerals and make iron and phosphorus accessible to life. And if you look at where iron and phosphorus come from out in the deep open ocean, wind-blown dust is a major source of iron and phosphorus. And so if you have this system here which is producing this excess DOC to stabilize iron, it may not be able to just increase the accessibility of that iron, may actually be able to increase the supply of iron and phosphorus to the open ocean. And of course, at the same time, as oxygenation proceeds, those negative feedback loops that I was talking about before start to fall away, and so the grip of iron, even though you're increasing, relatively speaking, the abundance of iron, it's still dropping, allowing it to, phosphorus to escape the grip of iron and increase in total abundance. There's an additional thing here, which is that the sort of conditions that are being promoted here, which is this dynamic in which you're drawing down the nitrogen, which you're increasing the bioavailability of phosphorus, of iron specifically, those are conditions that are favorable for nitrogen fixation. High iron, low nitrogen conditions are where you generally tend to see nitrogen fixation. And so it's rather interesting that molecular clocks suggest that the planktonic cyanobacteria that you see out in the open ocean emerge right around at the same time that the open ocean cyanobacteria do. So what this is then is sort of implying is that potentially the evolution and the expansion of these cyanobacteria out into the deep open ocean brought with it an increased supply of iron, phosphorus, and nitrogen out into the deep open ocean, stimulating total ocean ecosystem productivity. Now, if some of this sounds familiar to some people in the audience here, it's because people have actually suggested very similar mechanisms for what plants did when they colonized the continent. When plants experience nutrient limitation, they will excrete dissolved organic carbon from their roots, which dissolves the rocks and releases iron and phosphorus to become accessible to those plants, also stimulating nitrogen fixation in their roots. And this, was argued to be a key part of what allowed these systems to expand onto land, increasing the standing stock of carbon on land, and in that process driving oxygenation. And what I guess all I'm basically trying to say then is that you can argue a very similar kind of mechanism to what people have argued for plants happening out in the open ocean. And this is interesting because they happen sort of roughly in similar times in Earth history. This is happening somewhere in the Neoproterozoic towards the Phanerozoic boundary. This starts to switch on. This process starts to happen sometime in the early Phanerozoic, which is interesting because I was just trying to argue before that if you look at the metabolism of these systems, they look like convergent evolution, producing the same kind of organization of their metabolism, and specifically having a sort of a high metabolic rate type of phenotype that is part of this expansion. So it really starts to look then, if you kind of look at this level, that what's going on here is that you have now a process of convergent biogeochemical evolution where photosynthesis, oxygenic photosynthesis, is sort of expanding away from these sort of somewhat protective environments near continental boundaries where nutrient supplies may be a little bit more easier in some ways, out into the deep open ocean and out into the continents where nutrients are harder to access, and that has a key part to do with why oxygenation is happening in this time in Earth history. So stepping back then and looking at this sort of profile of Earth oxygenation, it becomes very tempting to speculate that what's going on here is that around this time is when you see first an expansion of self-damping cyanobacteria in sort of slightly more protective environments. But they're self-damping in that there's a limit to how much nutrients they can access. And only as they start to invent higher metabolic rate variants and these sort of higher power forms of oxygenic photosynthesis can life expand away into these sort of harsher environments, increasing the total standing stock of organic carbon and driving Earth oxygenation. And relating this back then to complexity also, and thinking about biospheric complexity and thinking about this in the context of increasing biospheric energy flux, right? Sort of around this time is when you sort of, a lot of evidence is suggesting this is when eukaryotes start to come on the scene, whereas after this period here is when you start to see multicellular, large multicellular organisms start to come online. So again, thinking about coupling the increase in mass, energy, and complexity of the biosphere. And so just to end then, which I always think is kind of fun to think about, is that if you start to think about the evolution of the biosphere through this lens, it's actually not all that different from thinking about how human societies have evolved in the post-industrial age, right? So as we have invented ways of pulling ever more electrons out of the Earth, buried electrons or electrons from plants and so on, we have increased the amount of artificial nitrogen fixation that we have been doing. We are increasing the amount of phosphate that we are mining from our minerals on Earth, and we are also increasing the rate at which we are extracting metals from our planet. And at the same time, the energy increases, not just the total energy consumption is not only increasing, but also per person is it increasing. And this is corresponding with the population boom of our human societies. Corresponding with all of that increased energy flow through the system, there's lots of innovations that are happening that are driving integration and the emergence of higher levels of organization with human societies as well. So our economies are ever more integrated, our social structures are becoming more integrated, and our political structures are becoming more integrated as well. So, it's sort of tempting then to think of the Industrial Revolution as a kind of a heterotrophic innovation in Earth history. And this is, of course, then also seen in the composition of the atmosphere, right? So, a lot of people focus on the increase of CO2 in the atmosphere, which, our burning of carbon is doing that. But actually, at the same time, we're drawing down oxygen, right? Through the burning of the carbon, we're drawing down the oxygen. Now, this is a very small amount on the total background of oxygen, so you don't really register it all that much. But nevertheless, it is going on. At the same time now, we're on the verge of a human autotrophic innovation, where we're sort of playing around with and trying to invent artificial photosynthesis. So I think it's sort of interesting to think about what this all means for the Earth system long term. This total increase in energy flux that we're pumping into the system, what will that do? So I'll leave it there and just put up the acknowledgments of all the people that have helped me in all of this and open it up for questions. Thank you.
Okay, questions for Rogier.
Thanks, that was a really interesting talk. I really like this idea of ecosystems being self-amplifying, self-organizing, evolving, trying to maximize, making a more energetic biosphere. Have you thought about what this implies for the emergence of life on other planets, specifically places like Mars, and the idea that maybe you could have a biosphere there that dies out, and what that means about the energy availability on other planets?
That's an interesting question. I have not myself thought about that very much, but that is an interesting question to think about. What are the available energy supplies and how long did they remain available?
Yeah, and what it implies about what a biosphere ending might look like.
Yeah.
Which is something that people hypothesize could have happened on Mars.
Right, yeah, it's a good question. I don't have very deep thoughts about that, but it's a good question.
Thanks.
Got to get Eric first to size hand. Long bomb.
That was lovely. My question could become irrelevant based on this first answer. Prochlorococcus don't fix nitrogen, correct?
Right. No.
So how does that fit into this? And maybe speculating why they don't fix nitrogen if they're optimizing rates of production. They've got all these free electrons.
Yeah.
Why not?
Yeah, it's a good question, and I can't actually say that I have immediately a very good answer to you, but it's kind of interesting that in general, nitrogen fixation and the major primary productivities are often separate processes. Same on land, right? Like their plants do the CO2 fixation, but it's then the microbes in their roots that are doing the nitrogen fixation. I don't know. I think it might have something to do with specialization and optimizing of the relative supply that you need of the two things, and how the problem is an ecosystem perhaps better solved by having more abundant of your primary producers and a slightly lower abundance of your nitrogen fixers, and that's sort of like a slightly more optimal solution in that sense. But it's a good question. I haven't really thought that through at the molecular sort of network level, how those trade-offs would come into play. But it is interesting that the two groups are expanding into the open ocean, sort of like kind of in the same time frame. So looking at that more closely and looking at their metabolic networks and at their distribution, so on, might be able to get at a better answer to your question. Right, so that's kind of what I was trying to say with the specialization, right? So Prochlorococcus is fiercely and furiously trying to optimize the acquisition of nutrients. And somehow, if you draw the nitrogen down to the lowest possible level, and you're best at that, that means that ultimately the ecosystem will supply the nitrogen to you, right? So the other cells will be doing the nitrogen fixation, and eventually it'll make its way back to you.
So that was fascinating.
Yes, exactly.
Let's keep going with the like that.
For the second oxygenation event,
yes.
The 1.5 billion year lag between the first and the second, is that essentially just waiting for the innovation of high-powered metabolism to come, or do you think there was another trigger that drove that?
That's actually a really great question, and something that I like to think about a lot, right? The question is, why is there this gap here? Why does one happen here, and then why does this happen here? Why doesn't it happen sooner? What is the reason for that? So one possibility could do that it has to do with the nature of innovation and the exploration of a mutational or the landscapes of things, something along those lines. I don't actually necessarily think that that's the main reason. I think there are potentially triggers in the Earth system that may have something to do with it. But exactly how that works, that's a very much of an open question. And so one thing that we need to do is to try to actually resolve the timescales of when these divergences of Prochlorococcus plus Synechococcus and the subgroups are happening, and sort of get that finer and try to line that up with what's going on in the Earth system at that time. But there's a few other things that are happening in the Earth system that might provide some constraints to where we should be looking for an answer. One is that there's a supercontinent Rodinia that's sort of breaking up right around this time, right? And this is, so these marine cyanobacteria are coming from freshwater ancestors, and the breakup of Rodinia is, of course, greatly expanding the sort of coastline space and the interface between freshwater and marine water. So it's maybe increasing the probabilities of going from freshwater to saltwater environments. There's also global glaciation events that go on in this time frame, where it's thought that ice sort of covers much of the planet, if not all. How does that relate to all of this? Is that potentially triggering some of these transitions, some of these evolutionary divergences, or is there some other causal mechanisms going on here? So I think by trying to actually integrate what we know from geology with what we see in the genomic record will help us constrain that question much better. Yes.
I'm interested in the oxygenation, but the events are episodic, and so you said they are, the new more efficient mechanism evolved and making a positive feedback. But we geologists said before the second rise of oxygen, billion years, nothing happened.
Right.
So my question is, what do you think prohibited the new innovations before these two events?
I guess in a way the specific question of what prevented the innovation is in a way I think similar to what we were just talking about, or what triggered the innovation. So it's a good question. I don't actually have a particularly good answer for you. But I should also mention about episodic and about oxygen rise. This is not somehow this sort of click, it switches on here, right? So you see actually perturbations in the carbon cycle that extend back to about 1 billion years or so before you really start to see the rise of oxygen. And then when oxygen starts to rise, it's not like it's suddenly at very high levels. It's new evidence sort of is continuously now suggesting that oxygen doesn't really start to rise towards modern levels until actually well after this boundary, at maybe somewhere between 300 and 400 million years or so. So really trying to understand what are the sequences of innovations, across that whole period, and how does that relate to what's going on in the Earth system, is a much better understanding of how the two line up are needed to be able to answer that question.
Well, thank you very much for the talk. I'm probably just going to go back to something that you said at the very beginning. I probably trying to capture again towards the end, I guess. Do you believe that, you talked a lot about positive and negative feedbacks actually as a possible way of not a possible way, as the way of interpreting what's going on in different phases. And yeah, you also mentioned that you're not you were not as close to the origins of life as you could have been. But do you believe that there is a chance we could be able to capture some sort of list of invariant properties related to either positive or negative feedback, or a combination of both, that will allow us to go back and say, well, yeah, once you find that sort of, interaction of whatever components we are talking about, then there is a chance that something interesting is emerging.
Yeah, so I don't know if this is answering 100% directly your question, but the question of trying to look for general principles that are coming from what we see in evolution and trying to think about emergence, I think this might be one place where we can think about that, right? Because what I've been trying to say here, or what I was trying to say here, is that this sort of excess electrons flowing through the system and causing a buildup of potential, essentially, that then leads to an emergence of a feedback loop between autotrophy and heterotrophy that sort of continues that process, but it's a sort of a higher level of organization of that kind of dynamic. That actually fits in a general sense with, some people like to think about the emergence of metabolism in very similar ways, right? Where you have an autocatalytic kind of proto-metabolism with ever longer self-amplifying feedback loops that sort of define the overall structure of metabolism, growing out and growing away from prebiotic chemistry. And so I would say that those kinds of thinking fit nicely with each other. And actually coming back to the discussion about autotrophy and heterotrophy, there's an interesting point there as well, right? Which is, in this case, I'm talking about innovations on the autotrophic side, but also on the heterotrophic side. And those need not happen ever concurrently. There may be delays between them, right? Just as we were talking about the emergence of RTCA and OTCA and autotrophy and heterotrophy and what is happening before the LUCA and so on, it could very well be, and in fact, I personally like the idea that in the evolution of these ecosystems and of the biosphere, that you have lags between autotrophic and heterotrophic innovations, and that there might be some connections between those lags and the perturbations that we see in the carbon cycle. Right, so you'll see periods in the carbon cycle where for tens of millions of years, maybe it sort of looks with a positive carbon isotope skew, and then there's a massive perturbation towards negative, then it goes back to positive. And so I think it's at least interesting to ponder whether there could be a role in all of that for delays between autotrophic and heterotrophic innovations. And given that in the extant biosphere, if there's any merit to such an idea, there appears possible at least to have substantial lags in time between autotrophic and heterotrophic innovations. That becomes interesting to think about then in the context of early ecosystems, and what kind of lag might you have between autotrophic and heterotrophic innovations, and what does that make us think about the boundary of the LUCA, for example?
Hi, thanks for the great talk. So my question is a little bit more on the kind of sociological, speculative side. So in particular about the very last part of your talk, which I find very interesting. So yeah, we've seen this very disturbing exponential growth in all kinds of sociological and sort of economic metrics, which
Right.
of course is not sustainable in the long term. And so, yeah, basically, if it carries on, the Earth collapses. But there is an alternative, which we see hints of right now, and that is if we, instead of generating tons of entropy from our sort of metabolic activity and traveling around using fossil fuels, etc., if instead we get our enrichment from just engaging with digital technology, if you extrapolate forward, you have this sort of dystopian vision where in the future, we're just kind of in the matrix, we're asleep all the time, being pumped with whatever nutrients we need, but we live our lives to the full in some digital world, because right now, we all kind of feed off social media, and social media feeds off content generated in the real world, but it wouldn't be difficult for all that content to be generated also in the digital world, at which point you don't need to engage with the real world at all. And so this sort of dire vision, on the one hand, it's a bit scary, but it does solve this I mean, the entropy produced from just manipulating all those bits would probably be less than like 10 billion people flying all around the world and that kind of thing.
Right.
So it might save planet Earth, and it might also explain Fermi's paradox.
Right. So lots there. I think speculation on that end is a little bit beyond me to be able to judge that. But there is an interesting point actually coming back to what we were just talking about, general ideas and general principles. And you know what I was trying to think about or trying to argue here, and maybe this relates a little bit to what you're talking about, is these higher levels of organization that start to emerge and increase metabolic rate at the ecosystem level. There's actually an interesting way to think here about the balance between maximizing rate and maximizing efficiency, right? So overall, you're still maximizing rate, but these innovations that produce higher levels of organization are in a way increasing the efficiency of the system, right? And so that then drives down the amount of waste that you're producing and so on. And so, you know, perhaps in thinking about human societies, things like the emergence of recycling, for example, would be one example of that. But so maybe, you know, our complexification, our sort of our social systems, maybe that increases our efficiency actually as well. So
Yeah.
All right, so we're at time. So let's thank Rogier again for his great talk.